हेलो फ्रेंड्स, वेलकम बैक टू अवर न्यू वीडियो। आज हम इस वीडियो में स्टार्ट करेंगे एमसीक्यूज ऑन बायोटेक्नोलॉजी। ओके, एंड दिस इज द फर्स्ट वीडियो ऑफ दिस सीरीज ऑफ वीडियोस ऑन एमसीक्यूज ऑन बायोटेक्नोलॉजी। ओके, सो let's start. So the first question of this video is, what is C value stands for? And the C value means, the options are the total amount of DNA in diploid genome, or option B, the total amount of DNA in triploid genome, or option C, the total amount of DNA in haploid genome, or the total amount of DNA in tetraploid genome. So the right answer about the C value is option C, that is the total amount of DNA in haploid genome stands for C value. Okay, so let's move on to the next question of this video, that is Bacillus thuringiensis was firstly discovered by bacteriologist option A, Ischiotta Sigetani, or Ernest Berliner, or Dr. Scotch, that is Robert Scotch, or F. Kohn, that is Ferdinand Kohn. Okay, so the right answer about the Bacillus thuringiensis was firstly discovered by bacteriologist, that is option A, that is Ishiwata Shigetani. Okay, so let's move on to the third question of this video. So which of the following mutation is ineffective for length concern? So which is ineffective for length of the polypeptide? And the options are simple mutation, missense mutation, or option C, nonsense mutation, option D, no such mutation exists. And the right answer about this is option B, that is missense mutation. So missense mutation is ineffective for length concern. Okay, so let's move on to the number 4. Question number 4 is, which of the following is or are concerned with the phenomenon of excision repair? And the right answer and the options are ligation only, option B excision only, option C incision only, option D all of the above. And the right answer is option D, that is all of the above. So all of the above, that means ligation, excision and incision, all are concerned with the phenomenon of excision repair system. Okay, so let's move on to the question number 5, that is strategically how many steps are there in PCR process? And the options are option A 5, option B 10, option C 3, option D 2, and the right answer is option C, that is 3. So strategically 3 steps are there in PCR process. So move on to the next one, that is when PCR sequencing is done for rRNA, the concern is about option A, its complementary strand, option B, certain phylogenetic relationship, option C, the coding DNA it has, and option D, it is not performed for rRNA. And the right answer is option B, that is certain phylogenetic relationship. So when PCR sequencing is done for rRNA, the concern is about certain phylogenetic relationship. Okay, so let's move on to the question number 7, that is improved varieties of wheat suitable for Indian climate have been developed by option A, hybridization and mutation, option B, mutation and cloning, option C, cloning of polyploids, or option D, polyploidy and hybridization. And the right answer is option A, that is hybridization and mutation. So improved varieties of wheat suitable for Indian climates have been developed by hybridization and mutation. So let's move on to the next question, that is both the environment and the genotype are important in the development of option A disease, option B country, option C plant quarantine, or option D, phenotype. And the right answer is option D, that is phenotype. So both the environment and the genotype are important in the development of phenotype. Okay. So next question देखेंगे. Next question है question number 9. So which of the following gene therapy vectors preferentially infect nerve cells? And the options are option A, adeno associated virus, option B, retrovirus, option C, herpes virus, option D, adenovirus. And the right one is option C, that is herpes virus. So herpes virus, so which of the following gene therapy vectors preferentially infect nerve cell? That is herpes virus. So herpes virus vectors preferentially infect nerve cells. Okay, so let's move on to the question number 10. That is basically, Bacillus thuringiensis was first named by Acibota Sigetani or Ernest Berliner, or option C, Dr. Scotch, option D, Ferdinand Kohn. And the right one is option B, that is Ernest Berliner. Okay, so let's move on to the next questions, that is question number 11. So, which of the following is or are different forms of DNA? And the options are option A, bDNA, option B, aDNA, option C, zDNA, option D, any of the above. So, the right answer is option D. That is any of the above. So, any of the above, that means bDNA, aDNA, and zDNA all are different forms of DNA. Okay. So, next question क्या है? Next is question number 12. The term back mutation stands for option A, reverse the effect of a mutation that had inactivated gene and thus restored, or option B, that is simultaneous mutation treatment. Option C, mutation is backside of gene. Option D, no such concept exists. So the right answer about the term back mutation is option A, that is reverse the effect of a mutation that had inactivated gene and thus restored. Okay, so let's move on to the next, that is question number 13. So biological vectors are used by scientists for the purpose of to carry new gene into cell, or option B, to bring genetic addition to recipient, option C, for expression of novel characters, option D, all of the above. And the right one is option D, that is all of the above. So all of the above, that means to carry new gene into cell, to bring genetic addition to recipient, for expression of novel characters, or are the purpose of biological vectors. Biological vectors are used by scientists for the purpose of these three reasons. Okay, so next question देखेंगे, question number 14. So what is beta carotene? So option A is, it is a phytochemical naturally produced in carrot and orange vegetables, or option B, mutant carotene protein, or option C, artificial protein, option D, none of the above. So the right one is option A, that is it is a phytochemical naturally produced in carrot and orange vegetables. So beta carotene is a phytochemical naturally produced in carrot and orange vegetables. Okay, so question number 15 देखेंगे. So which of the following is or beta-lactam antibiotics? And the options are penicillin G or ampicillin, or both A and B. That means both penicillin G and ampicillin, or option D, that is neither A nor B. So the right answer is option C, that is both A and B. So both A and B, that means penicillin G and ampicillin, are beta-lactam antibiotics. Okay. So let's move on to the next question, that is question number 16. So what do you mean by beta cells? And the options are B line of cell culture, option B, insulin producing cells in pancreas, option C, cells antagonistic to alpha cells, or option D, cancerous cells. So the right answer is option B, that is insulin producing cell in pancreas. So the beta cell means that are the insulin producing cells in pancreas. Okay, so question number 17 क्या है देखेंगे. So question number 17 is biochip is a device that utilizes biological molecules as framework, gives applications such as option A analyze very small sample of DNA, option B assess impact of drugs, option C detect pathogen or cancerous cell in blood sample, or all of the above. And the right answer is option D, that is all of the above are correct. So biochip is a device that utilizes biological molecules as framework, gives applications such as analyze very small sample of DNA, assess impact of drug, and detect pathogens or cancerous cells in blood samples. Okay, so let's move on to the question number 18, that is what the term biocide stands for, and the options are any drug or chemical detritus to non-living agents, or option B any drug or chemical detritus to non-pharmaceuticals, or option C any drug or chemical detritus to living agents, or option D any drug or chemical used to cure bioweapons. And the right answer is option C, that is any drug or chemical detritus to living agent is known as biocides. Okay, so question number 19 देखेंगे. So what do you mean by the term biolistic? And the options are option A used to shoot pellets coated with genes into certain living structure. Option B, it is a biotech fantasy. Option C, used to cure biotechnological losses, or option C, no such word exists. And the right one is option A. It is biolistic is used to shoot pellets coated with genes into certain living structure. So the last question of this video is a polyploid with genomes derived from same original species is known as amphidiploid or alloploid or autoploidy or autoploid, autopolyploid or autoallopolyploid. So the right answer is option C, that is autopolyploid. So a polyploid with genomes derived from same original species is autopolyploid. Okay, so these are all 20 questions of this video on MCQs of biotechnology. Okay, so thank you for watching this video.